In bacteria, the third stage of protein synthesis is elongation. The requirements of elongation are initiation complex, amino acid tRNAs, GTP, and elongation factors. The elongation factors are EFTU, EFTS, EFG. Before elongation, an initiation complex is found in the bacterial cytosol. In bacteria, the elongation stage of protein synthesis has three steps. In elongation step 1, the appropriate amino acid tRNA binds to EFTU GTP complex. As a result, amino acid tRNA EFTU GTP complex is formed. The amino acid tRNA EFTU GTP complex then binds to the A site or amino acid site of 70S initiation complex. The GTP bound to EFTU then undergoes hydrolysis to produce EFTU GDP complex and PI. The EFTU GDP complex and PI are then released from the 70S ribosome. The EFTU GDP complex then binds to EFTS. The GDP bound to EFTU is then released. As a result, EFTU EFTS complex is formed. Another molecule of GTP then binds to EFTU. The EFTS is then released. As a result, EFTU GTP complex is formed. This EFTU GTP complex is now available to repeat the cycle. Now, the P site or peptidyl site has FMAT tRNA FMAT and the A site has amino acid tRNA 2. In the elongation step 2, the first peptide bond is formed in presence of peptidyl transferase. The peptidyl transferase is a 23S rRNA ribozyme. Means the peptide bond formation reaction is catalyzed by the 23S rRNA. The peptidyl transferase is present in the 50S ribosomal subunit of bacteria. During peptide bond formation in presence of peptidyl transferase, the nitrogen present in the alpha amino group of amino acid tRNA uses its lone pair of electrons to attack nucleophilically to the carbonyl carbon of the peptidyl tRNA. The electron pair is then transferred from carbonyl carbon to the oxygen atom. As a result, a peptide bond is formed. The ester bond linking the peptide to the 3' oxygen of peptidyl tRNA breaks, leaving a deacylated tRNA. During peptide bond formation, amino acid 1 is transferred to amino acid 2. As previously said, due to the peptide bond formation, a deacylated tRNA FMAT is formed in the P site. Also due to peptide bond formation, a dipeptidyl tRNA 2 is also formed in the A site. In the elongation step 3, an elongation factor called EFG or translocase binds to the GTP. The EFG GTP complex then binds to the ribosome. The GTP bound to EFG then undergoes hydrolysis to produce GDP and pi. The pi is then released. The energy released during the GTP hydrolysis causes the movement of ribosome along the mRNA. This movement of ribosome is called as translocation. The structure of EFG mimics the structure of EFTU tRNA complex. So, EFG can bind the A site and presumably displace the peptidyl tRNA. As shown here, after translocation, EFG GDP complex occupies A site, dipeptidyl tRNA2 occupies the P site, and the deacylated tRNA FMET occupies the E site or exit site. EFG GDP complex then leaves its occupied site in the ribosome. An amino acid tRNA3 then occupies the A site in the ribosome. At the same time, the deacylated tRNA FMET leaves the E site. Now, the ribosome forms another peptide bond between the second amino acid of the dipeptide and amino acid 3. The dipeptidyl tRNA2 in the P site of ribosome looks like this. Now, some important points. Eukaryotic ribosomes do not have an E site. In eukaryotes, uncharged tRNAs are expelled directly from the P site. Prokaryotic EFTU is functionally analogous to eukaryotic EEF1 alpha. Prokaryotic EFTS is functionally analogous to eukaryotic EEF1 beta gamma. Prokaryotic EFG is functionally analogous to EEF2. Please like, subscribe and share.